Now come to the next, that is acquired immunity is pathogen specific and characterized by the memory and the both or the non-specific. Acquired immunity actually it is pathogen specific as well as characterized by the memory. Why? Listen carefully. When we talk about the acquired immunity, when pathogen enters in the body, right, what will happen? If that pathogen, we use the chemical of that pathogen and form the antibodies against it, right? And if we already aware with that pathogen, the nature of that pathogen chemical, right, then we already have the antibodies against them, right? And what will happen now? In that case, we recognize the chemical antigen of that particular pathogen and search in our memory cells that if this pathogen is known to us or it is unknown. If it is known, that means it's antibodies against the it's against it, the antibodies is already present in our body, right? So acquired immunity is based on the pathogen specific and the characterized by the memory both, right? So the answer is C. See here what will happen. When we talk about the acquired immunity is also known as the specific immunity, right? That acquired immunity is also known as the specific immunity. Clear? Now what will happen in that case? When pathogen enters first time in our body, when pathogen enters first time in the body, right, then what will happen? Our body responds with low intensity. Why? Because it is new for us, right? Low intensity because we don't know about the power of the that pathogen, we don't know about the chemical nature, we don't know how we destroy it, right? So the first response is with low intensity, right? But in that case, with the low intensity, when we recognize the chemical nature of that, right, so the primary response is the low intensity, right? The primary response is of the low intensity. But what will happen? Even after that, some pathogens encountered by our body cells, right, encountered by the body, right, and what will happen in that case? They recognize its chemical nature and form the antibodies against them by the use of its own antigen, right? And what will happen now? It creates the high response, right? It creates the high response, right? And in NCRT, one word is used there, that is intensify. Intensify secondary of secondary of the anamnestic response, right, which is also known as the secondary response actually. Clear? Which is also known as the secondary response. So in that manner that acquired immunity acts upon our body, right? When we talk about that, they are of two types. That acquired immunity are of two types. First one is the humoral immunity, which is also known as the antibody mediated immunity, right? Second is the cell mediated immunity, cell mediated immunity, which is known as the CMI, that is the cell mediated immunity, right? And for humoral immunity, B lymphocytes is responsible for their, and for cell mediated immunity, T lymphocytes are responsible for their. Clear? So in that way, it is characterized by the pathogen specific and characterized by the memory cells also. And one more thing, that these B lymphocytes when pathogen enters in our body, right, that these B lymphocytes actually, why they are called B lymphocytes? Because they gets mature and developed and mature in the bone marrow, right? And when they leave the bone marrow, each B lymphocyte express a unique antigen binding receptors on its membrane, right? So now what will happen here? B cell receptor is a membrane bounded antibody molecule, right, which is chemically a glycoprotein, binding of the antigen to the antibody, right? When binding of antigen to the antibody, it causes B cells to divide rapidly, right? And when these cells divide rapidly, what will happen? They form two types of cells, right? Progeny of B cells form two types of cell and they differentiated into two. First one is the memory B cell, right, which can retain the memory of the chemical nature of that particular pathogen, right? So when they if they enters second time in our body, so we have the memory cell, we can recognize the chemical nature, and B lymphocytes may prepare the antibodies against them and counteract them, right, and kill them. So in that manner. And the second is the effector B cell. Second is the effector B cell, which is also known as the plasma cells, and these plasma cells forms the antibodies, right? These plasma cells forms the antibodies. So in that manner, that's why we can say that The acquired immunity is the pathogen specific as well as the characterized by the memory, right? So now come to the next question. B lymphocytes and T lymphocytes comes under, we already studied here, right? They comes under the acquired immunity, right? And one more thing, when we talk about the T lymphocytes, they are of three types: T helper, T killer, which is also known as the natural killer cell in NCERT, and third one is the T suppressor. These T helper cells helps to recognize the chemical antigen to the B lymphocyte and helps in the formation of the antibody, right? And that T killer cell destroy the grafting tissues and foreign particles, and T suppressor cells suppress the overactivity of the T killer cell, so we can prevent our own cell by the destruction of T killer cells. Sometimes what will happen, to destroy the foreign foreign antigen or the foreign tissue, that T killer cells destroy our own cells also, right? So to suppress them, T suppressor acts upon it, right? So B lymphocyte and T lymphocyte comes under the acquired immunity, right? That is the according to the requirement our body can generate the antibodies, so that is the acquired immunity. Now come to the next. B lymphocytes are responsible for, we already discussed it, that B lymphocytes are responsible for the humoral immune response, and for cell mediated immunity, T cell lymphocytes are responsible for that, right? Now come to the next question. Each antibody has the 4 peptide chains, right? Each antibody has the 4 peptide, how many peptide chains? So each antibody has the 4 peptide chains, right? 2 heavy and 2 light. Each antibody has how many peptide chains? How we can say it is 4 peptide chain? Actually, see that is the structure of antibody, right? This is the structure of antibody. There are 4 peptide chains, that is 2 light chain and 2 heavy chains, right? That's why we say that the 4 peptide chains are there, right? See, that is the light chain, right, and the heavy chain. This is the constant region of the heavy chain, clear? That is the this is the antigen binding site where the antigen gets attached, right? And these chains are attached with the disulfide bond, right? These chains are attached with the disulfide bond. That fragment is the constant fragment. There is no change in that fragment. That's why it is called the constant fragment, right? Clear? So this is the structure of the antibody. Antibody releasing cells are, we know that the antibodies is secreted by the B lymphocytes, and specifically B lymphocytes, when B lymphocyte gets differentiate, they forms 2 types of cell. First one is the memory cell and second one is the effector B cell, or which is also known as the plasma cells, right? So we can say the plasma cells form the antibody exactly, and B lymphocyte also. So the answer is A and C, right? So the most appropriate correct answer is the D, that is A and C, right? Now come to the next question, that is 13th. Physical barrier, physiological barrier, cellular barrier, cytokine barriers, all these are the type of the innate immunity, right? They comes under the innate immunity, that is inborn immunity, clear? When we talk about the physical barrier, that physical barrier, that means directly physical barrier, that means no foreign pathogen enters directly inside our body, right? Clear? So that is first one. When we talk about that is the skin is the first line of the defense of our body, and it cannot permit the entry of the any foreign pathogen. Physiological barrier, that means by the functioning, that is by the secretion of the HCl, right, by the enzymatic action like the lysozyme. Cellular barriers in which cells are involved, right, cytokines barriers. So these are the innate immunity, right, inborn immunity. Now come to the match the columns. Physical barrier, that is the skin is related with the physical barrier, and lining of the respiratory, alimentary and the urinogenital tract is the physical barrier. Saliva in mouth, tears, macrophages, no. So A, 4 and 5, right, so it is given in A and C option, so B and D cancel, right. Physiological barrier, that means by the action, so WBCs is also acts as a physiological barrier, right, so B. Then interferon, no. Physiological barrier, that is acid in stomach, is also the example of the physiological barrier, right. No, WBC is the cellular barrier actually, right. So acid in stomach is the physiological barrier. Now saliva in mouth is the physiological barrier. Tears, that is the physiological barrier. Macrophages, no, these are also comes under the cellular barriers, right. So B, 3, 6 and 7, B, 1 and 8, no, 3, 6 and 7. So the answer is C. A is also cancelled, right. Then when we talk about the cellular barriers, so what are the cellular barriers? Cellular barriers are the WBCs and the macrophages, right, and cytokine barrier, that is the interferon, right, cytokine barrier, that is the interferon. So these are the barriers which represents the innate immunity and prevent from the entry of the any foreign pathogen inside the body, right. Come to the next. Level of which one of the following increases in allergy, right. These, what are these? These are the types of the immunoglobulins, that is the antibodies, right. That Ig represents the immunoglobulin, right, it represents the immunoglobulin, and that immunoglobulin is related with the antibodies, right. These are the antibodies, majorly 5 types of antibodies we will have to study: IgA, IgG, IgM, IgE, and one more, that is IgD here, right. That IgA is present maximum in secretions of the body, right. That IgG is present maximum in amount, maximum in amount. 80% antibody, 80% is the IgG, right, and it is the only antibody which can cross the placenta, right. It is the only antibody which can cross the placenta. Clear? IgM, that is macroimmunoglobulin, that is responsible for, it is most efficient in agglutination, right? It is most efficient in agglutination, right? And now IgE, that IgE is concentrated in mucous membranes, skin and the lungs, right? And in case of any kind of allergy, these are the antibodies which is present in that case maximum amount. If they are increased in maximum amount, then also allergy is causes, and if some allergen enters inside the body, then that antibody against act on them. Clear? So the answer of that question, level of which one of the following increases in case of allergy? So answer is the IgE, right? This is all about the antibodies. Come to the next question, that is, which drug are used against the allergy? When we talk about the drugs which are used against the allergy, in case of allergy, first we use the antihistaminic drugs, which are the anti-allergic, right? Actually, it is the inflammatory histamine. We know that the histamine is the inflammatory, right? And what will happen? Inflammatory as well as the vasodilator, right? So in case this is actually the exact line of NCERT. In NCERT, that line is given, right? The allergy is due to the release of the chemicals like the histamine and the serotonin, right? So to reduce the allergic condition, we have to use the antihistaminic drug, right? The adrenaline as well as the steroids, right? So the answer is D. Answer is D. Clear? The quick acting immunity is active immunity, passive immunity, both and none. Now the question is, what is active immunity and what is the passive immunity, right? What is the difference between the active immunity and the passive immunity? Actually, these are the part of the humoral immunity as well, humoral immunity, right? Active immunity, that means it is mediated by the memory cells, right? It is mediated by the memory cells, and what will happen? Resistance built up as a result of the body's contact with the microorganisms and their toxins, which is known as the active immunity, right? But when in case of the passive immunity, we directly transfer the actively formed antibodies, right, from one person to another, right? That's why the quick acting immunity is the passive immunity as compared to the active immunity. Clear? Come to the next. Colostrum is rich in, we already studied that the in maximum secretions of the body, IgA is present in maximum amount, right? So colostrum, what is colostrum? That is the first milk after the birth, that is first pre-milk after the birth or after the parturition, right? From which is secreted by the mother's mammary gland. It is yellowish in appearance due to the high concentration of the antibodies, right? And it's very very important for the child to produce the innate immunity, right? So the IgA is rich in colostrum. Come to the next. Rheumatoid arthritis. That rheumatoid arthritis is an example of the autoimmune disorder, right? It's not related with the bacterial diseases, it's not related with the viral, it's not related with the other, right? But it is the autoimmune disorder. But yes, sometimes after the in some patients, in some cases, the consequences of the pneumonia, sometimes the rheumatic arthritis can be seen, right? But it is the autoimmune disorder. It's not related with the any kind of the bacterial disease or the viral disease. Yes, it can, the consequences of any kind of that disease, that's a different thing, but itself it is the autoimmune disorder, right? And in rheumatoid arthritis, what will happen, that is the swelling of the synovial joints, right? Clear. Synovial, there is the inflammation in the synovial membrane and of the long bones, right? So these the patient may suffer from the joint pain, right? The patient suffer from the joint pain, and in that case, in the rheumatoid arthritis case, what will happen, patient in patient blood, the rheumatic arthritis factor, RA factor, is comes in the blood, right? And when we detect it, so we treat them accordingly, right? There is inflammation, burning, redness, pain, swelling, so patient may suffer from the movement. Even movement of the body increases the pain of the arthritis, right? From the list of the site given above, select the ones which immature lymphocytes differentiate into antigen sensitive lymphocytes. Okay, antigen sensitive lymphocytes, bone marrow produce the immature lymphocytes, antigen sensitive lymphocyte, and even thymus. But rest of these, because both of them are the primary lymphoid organs, right? Bone marrow and thymus is the primary lymphoid organs which differentiate to the antigen sensitive lymphocytes. But the lymph nodes, the tonsils, spleen, Peyer's patches, all these are the secondary lymphoid organs, right? All these are the secondary lymphoid organ. Clear? So the answer is 1 and 4, that is C. Clear? Come to the next question, that is malt. Malt, what is malt? The full form of malt is the mucosal associated lymphoid tissue, right? Mucosal associated lymphoid tissue. Now actually, the what is that malt? This is the lymphoid tissue which is present in the within the lining of the major tract, that is the respiratory tract, the digestive tract, the reproductive tract, right? That is called the malt, or the urinogenital tract. So the answer in that part, the answer is the D, right? Actually, all the some lymph nodes are present in the body, and 50% of the lymphoid tissue present in the human body, right? So the next question is, AIDS was first time reported in, right? AIDS, that is acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome, right? Acquired immunodeficiency syndrome, it is first time reported in 1981, right? First time reported in 1981 in USA actually, right? The maximum cases, the 80% of the AIDS cases from whole worldwide, it is present in the USA just because of their lifestyle of them, right? Just because of the lifestyle of them. AIDS, actually, what is that AIDS? AIDS is caused by the HIV virus, right? It is caused by the HIV virus, right? HIV, that is human immunodeficiency virus. What will happen in that case? That HIV attacks on the T helper cells, right? How? See that AIDS was first time reported in USA, right, in 1981. Actually, its causative organism is the HIV, right, and which is identified by the Luc Montagnier. When we talk about that, HIV are of two type, right, that is HIV-1, HIV-2, right, and the most common infection caused by the HIV-1, right, that is the virus which is found in the blood of the wild African green monkey, which replicates to form the DNA, right, in which replicates to form the DNA. And what will happen, that HIV produces the DNA from its RNA, right, and then disintegrated into host cells DNA, right, and then disintegrated into the host cell DNA. So macrophages continuously to produce the virus instead of to kill them. That's why these kinds of macrophages is called the HIV factory. What will happen? It uses the host RNA to form the DNA template, and once they form the DNA template, they embedded that DNA template into the DNA, right? So it's not the now it is not the foreign DNA for our body. So what will happen, our body produce that in excess amount, right? And now what will happen simultaneously, that HIV also infects the T helper cells, right? Simultaneously that HIV also infects, that is, it causes infection in T helper cells, T helper lymphocytes specifically, right? And that T helper lymphocytes have the CD4 receptors, right? They have the CD4 receptors. This is the structure of the HIV. You can see these are the receptors, and these are the glycoprotein GP120. That GP120 gets attached with the CD4 receptor site, right, and destroy the cell completely. Clear? In 2016, the question asked upon the reverse transcriptase, how many reverse transcriptase are present in the HIV virus. So two reverse transcriptase are present in the HIV virus. Memorize it, right? This is the protease, this is the phospholipid envelope, that is capsid protein, right, that is 24. So simply when we talk about that, so that GP120 attach on that CD4 receptor site, and what will happen now, that HIV progressively destroys the T helper lymphocyte, right? That HIV progressively destroy when GP120 attach to CD4 receptor site, then HIV destroys the T lymphocytes, right? Now what will happen, if the T helper lymphocytes decreases in amount, then it will be difficult to recognize the foreign antigen for the B lymphocyte, and if it is not here, then B lymphocyte is not able to produce the antibodies against them, right? So the HIV destroys the T lymphocytes and activated the other lymphocytes also, right? And what will happen in that manner, our immune system gets collapsed completely, right? Immune system collapsed completely, right? And the uncontrolled infections enters in the body, right? What are the sign and symptoms of that? So we can say first we can see here, right, that retrovirus HIV virus, it uses our RNA to form a DNA template, right? It is the viral protein code. What will happen, that virus infect the normal cell, and when it infect the animal cell, normal cell, what will happen, that viral RNA is introduced in the cell. So viral DNA is produced by the reverse transcriptase, right? Viral DNA is produced by the reverse transcriptase, and now that DNA, that viral DNA, that is incorporates with the human genome. So what will happen now, when it incorporates with the human genome, then new viral RNA is produced by the infected cell. And what will happen after that, the all cells are the infected cells which are produced by them, right? So all this is the action of the HIV virus on our body. And when we talk about the sign and symptoms, actually its incubation period is so much long, right? The clinical symptoms of AIDS usually appear when the T lymphocytes level below than the 200 per millimeter square of the blood, right? Per cubic mm of the blood, then first time symptoms appear. So what are these symptoms, right? These swollen lymph nodes, mild prolonged fever, diarrhea, pneumocystis, carinii, pneumonia, Kaposi sarcoma, that is a type of skin cancer, drenching sweat, night sweat, right? So these are the symptoms of the AIDS, right? The World's AIDS Day was celebrated on the 1st of December, right? When we talk about its symptoms and also how to diagnose the AIDS, some processes are used to diagnose it, right? Some kind of test, that is diagnostic test, for the AIDS. First is the ELISA or the ELISA, that is the, what is the full form of that? Enzyme linked immunosorbent assay, right? Enzyme linked immunosorbent assay. This test is confirmed by the Western blot test, right? This test is confirmed by the Western blot test. Aura quick diagnostic device is used to detect the HIV virus within 20 minutes, right? When we talk about after the diagnosis, right, in treatment, we use the AZT, that is azidothiamidine, right? That is azidothiamidine, which is also known as the retrovir, right? Clear? So in that way, and AIDS day is the 1st December. Clear? So this is all about the AIDS, and its detail is given in NCERT, right? And it's very important from the examination point of view, so you have to learn it properly, right?